Thank you.
Good, how are you?
So you're on the call, correct?
We did meet on the call.
We're good to go. Yeah.
Is it hot enough for you?
Man.
Yeah, it is hot.
Yeah. So, if there are any like scientific words that come up,
It looks like I'm struggling.
We need to look like we're doing something
So
productive.
this microphone goes to the room. I'm gonna try and call folks right here. This is good. This goes to
that's really interesting.
Excuse me, we're gonna get started in about a minute here. If people can take their seats. Thank you. We have a new microphone. Can everybody hear this okay? All right, welcome everybody, Lou Casa, to our 2022 Entrepreneur Spotlight, basically our Shark Tank, but for copyright reasons, we'll call it that. We've got three great new companies here. We've got three great panelists, our sharks. And then, of course, afterwards, and I hope everyone could stay, we have a really special Wall of Fame presentation for Senator Steve Sanacero. And then we'll have a reception and networking afterwards. Before I introduce our MC, first time up here without a very special partner who's usually up here when we do these introductions. So I would like to, if everybody could just pay a special tribute to Tim Block, our President Emeritus, who's here in attendance. His farewell tour continues, and now I really miss having Tim up here, and he transitioned to our chairman June 30th, and it's great to have Tim here. I'm going to introduce our MC. He's been doing this for quite a bit. I don't know how many years, Grant, is this 5, 6, and he does just a tremendous job. He puts a lot of energy, a lot of time into it. Known Grant now for probably 5, 6 years. He's an ambassador for the Pennsylvania Biotechnology Center. And some of you may or may not know, he's got a tremendous career as a VP of Commercial Operations for Sentinel Therapeutics. And that's an international and national business development. He's kind of a jack of all trades, fireman in Doylestown. And recently, because I have to say one new thing, because I've done your intro so many times, we were out in San Diego for Bio, and they do these really cool thing now where they do a 5K before you go and do the conference. So I told Grant about it. He just recently got into running, and lo and behold, on the San Diego Bay Harbor, he set his personal record best time. So, ladies and gentlemen, Grant Castor, thanks.
Cool. And just to set the record clear, he's still a much faster runner than I am. So I should have put my name on the back of your shirt through it. So anyway, welcome to the 8th annual Entrepreneur Spotlight. So over the next 1.5 hours, we'll have 3 unique opportunities presented to us by 3 passionate entrepreneurs. All right. So before I begin, and should I step back a little bit? I don't know if this microphone sounds a little muffled. All right. I have the, before I introduce our distinguished panel, I'd like to give a brief update. We good?
Yeah.
All right, cool. A brief update on what has happened to the biotech market in the last 12 months. So everybody's sitting down, right? You better hold on to your chairs. All right. So I read quite a bit. And I was reading an analyst report this past week, and I think one analyst said it quite right. We have seen essentially two weather fronts. We've seen a nor'easter collide with an Arctic front. So the Arctic front can be defined as inflation, while the nor'easter can be defined as the positive money flow into the sector. So the two are absolutely clashing in what has become a very liquid biotech sector. So some not so fun facts to kick things off. Biotech enterprise values are down 64% since peak and 41% year to date. There were 183 life science companies last week that had a negative enterprise value. And that number is down slightly from approximately 220 in mid-May. The market has become more discerning since the start of the year. The premium for a very good phase 3 data set has barely dropped. In contrast, if you have a somewhat good, a medium, or a poor data set, those organizations have seen their valuations cut in more than half year to date. All right, so enough with the negative. Where's the opportunity? If you're on the buying side, which I am, there are some great values to be sought in the current market. When do you remember greater than 300 companies trading or having more cash on their balance sheet than their market cap? So quite an interesting dilemma right now. If you're on the capital raising side, now is a great time to dip into private vehicles and spend time making your TPP or target product profile and business strategy succinct and distinct. In my opinion, we're already in a recession, just my opinion, and the Fed will continue to be hawkish, which will in turn result in Paul Volcker-like action. I wasn't born then, but history always repeats itself. While this might sound negative, I view this as true opportunity. Typically, those that can hunker down, be scrappy, focus on what is impactful, and execute outperform those that throw 100 targets at a wall while bleeding capital, praying that something will stick. Then to realize when something sticks, that they've run out of capital and are deep in the valley of death, or what I call the pit of misery, dilly dilly. All right, so last week I was invited to speak at the World Orphan Drug Congress up in Boston. While I was there, I had an opportunity to attend a session on rare disease. And during that session, it was mentioned that it's now believed that there are over 11,000 distinct rare diseases. If for those of you familiar, NORD is a really great advocacy group, and they've historically said there's about 7,000 defined rare diseases. So if that number is indeed true, that's a 40% increase in defined diseases in just the last few years. So what does that mean? Oh, to me, we as entrepreneurs, drug developers, and investors have a huge opportunity to close the gap of therapeutic options for those diseases, as only 5 to 8% of the known diseases have a therapeutic option. Rare disease is not the only segment in this diverse market in which we are able to tailor our therapies in more specific ways that can result in significant upside for both patients and us as entrepreneurs. So with that, I'd like to introduce our distinguished panel. All the way to the right, we have Rose McDevitt. She's a venture capital analyst with Ben Franklin Technology Partners. To the left, Bill White, head of North American Strategic Partnerships with Janssen Pharmaceuticals. And in the center, Matthew Burkhardt, director of B+ Labs, my GPA, and Sierra Center. Okay, so quick housekeeping items before we begin. The presentations will be approximately 10 minutes, timed by the traffic light. Please hold it up, so keep everybody honest. Followed by approximately 5 minutes of questions from the panel and the audience, and I guess online as well. And then most importantly, please join us for a reception and opportunity to meet with each entrepreneur after our conclusion here approximately at 5 p.m. So with that, I'll turn it to Rose to introduce our first opportunity and entrepreneur. Thank you.
All right, so I'll start by presenting NeuroPathX. NeuroPathX is a biopharmaceutical company developing next generation of therapeutics for pain management and looking to address the opioid crisis. The company has developed a proprietary cannabinoid derivative compound as a lead drug molecule to treat neuropathic pain and address the opioid crisis. Their patented family of monotherapeutic compounds are focused on treating a variety of diseases, That includes oxidative stress-related diseases, neurodegenerative disorders, and neurodegenerative disorders and chronic pain management. The technology definitely has the potential to address a significant unmet medical need. And presenting the technology today is Dean Petkanas. Dean has over 25 years of experience in executive management, investment banking, and corporate finance. And he is also the co-founder, co-inventor, and CEO of NeuroPathX. Dean, I'll turn it over to you.
Thanks a lot, Rose. Thank you, everybody, for being here. It's a real pleasure and an honor to be presenting the company here. And congratulations to Lou and the Biotechnology Center for successful completion. It's a beautiful facility. Shout out to my team members, Bill Kinney, Doug Brenneman, Mark McDonald, Terry Tormey, Bill, Doug, and Mark, all Jansen guys. So I know Jansen's in the house here today. I hate buzzwords like crisis and fundamental and stuff like that, but there's a real crisis in America today, and it's an opiate crisis, and it's not getting smaller, it's getting larger and larger. And the fact of the matter remains is that there's a supply chain of opiates in the market that were used for many, many years to treat a variety of different pain-related disorders, from analgesic to neuropathic pain. I call it Ben Johnson on steroids out of the running blocks. And what had happened over the years is Big Pharma got routed out of CNS-related drugs, and hence the opportunity now exists to take down a mountain of an oversupply of opiates in the marketplace. We're pioneers in the area of cannabinoid therapeutics. We started about 12 years ago looking at CBD as a primary candidate to deal with a variety of different disorders, one of which was neuroprotection. We found that there was some aptitude in CBD in being a neuroprotectant, and we shifted gears very, very quickly through a grant study that we did with Temple University, and we got into the field of neuropathic pain. We compared our compound to CBD to look at pharmacokinetic design and the efficiencies and inefficiencies of that compound. There was a lot of promise in CBD, but we very quickly unearthed some of the dark side issues of cannabidiol, and we'll get into that in a little bit. Backbone of our science is neuroprotection. It focuses on reducing inflammation. Inflammation is becoming a buzzword in the field of medicine these days.
Can't sneak in.
Okay. I'd rather hear my dog barking. One of the great points of our technology to date and the success of our preclinical discovery in MedChem and our pharmacology has been the receipt of a $2.97 million grant from NIH, highly leveraged, capable money right now. So if you're investors out there listening, we could use you today to leverage against IND enabling studies that we have. We have 5 peer-reviewed papers and publications, including the latest in the Journal of Neuropathic Molecular Neurosciences and in the British Journal of Pharmacology last year. Very important, opiates used to treat chemotherapy-induced neuropathic pain. That's our first target, and there's no FDA-approved drug right now to deal with that problem. So there's your market right there. This is the big problem. It's pain, and pain leads to addiction. You could see the numbers are startling, and they're not going down, they're actually going up. And the average care cost per individual is going up as well, and it's a huge burden on society. I'll just give you a little sidebar of some knowledge that I have. The DEA is currently in place at API manufacturers that are selling hydrocodone, oxycodone, OxyContin to compound pharmacies, and they're being stopped from delivering products to that market. So you can see there's a changing dynamic underneath it all, and opiates are being rooted out of the market. I think that there's a useful model for opiates. It's in palliative and end-of-life care, and maybe initially after post-op, and it should be under the care and provision of those that are skilled in the art of delivering the medicament. So that's another side of the market there. There's obviously there's a use of off-label for opiates. It's time for us to end this crisis. That's a big mountain right there. There's a huge opportunity. 29.8 billion in a replacement market seems to me like a good place to be. Big Pharma is jumping back into pain. People tell you that they're not interested. That's a lot of hogwash. I can't tell you who we're talking to, but we are talking to Big Pharma. One is in pain and another is in a G protein coupled receptor space, which we'll get into. Other markets we could jump into are diabetic neuropathy, post-herpetic neuralgia, and opiate abuse disorder. And our target compound, our lead compound, has target efficiency in areas of inflammation and also the reduction and withdrawal of opiate and morphine dependency. This is our lead compound. Kudos to Bill Kinney, Mark McDonald, and Doug Brenneman for utilizing this competency of drug discovery and pharmacology. We basically recrafted some of the design of CBD. At the very end of that compound, you'll see there's a funny little upward moving tail. That's an azetidine chain. It's off of a pentyl group. That compound was changed to change the inefficiencies associated with CBD. Poor bioavailability, lipophilicity, not aqueous insoluble. So you're looking at a novel breakthrough compound. We've gone a real far way from 2013, a 7-step low yield is now a 4-step high yield at 25 grams. And our gratitude goes out to Purus, a former Aramco and Janssen Group, for having modeled up the scale-up design. Next year, we'll go to 200 grams scale-up. Our preclinical results are all in the CIP and animal model right now, and some other models that the preclinical screening program for pain over at NIH is doing for us in other areas, like screening for anti-seizure or anticonvulsant, screening for osteoarthritis in a tier 3 study under the PSPP, and we have some other things that are going on in neurodegen as well. Our targets are GPR55, which regulate NLRP3 expression. You guys know anything about inflammation, you know that that's a centerpiece and a big money ball. Roche bought out Inflazome in 2020 for $447 down and up to $2 billion in milestones. And Eli Lilly bought Disarm in the same year in October, also targeting NLRP3 as an inflammation model. But both of those were acquisitions done for the CNS. I'm glad to say here today that we're in both systems, in the PNS and the CNS. We also have shown in our preclinical studies to reduce morphine reinstatement in the animal model. Both these targets, NCX1 and GPR55, are implicated. I'll show you a slide in a little bit, but we have some validation in both targets. So our product is, is a potential platform. It's not to throw everything up against the wall and hope to spray and pray. We took a traditional pharma approach to drug discovery and pharmacology. We know that we had something in hand. We worked on it. We tried to perfect it. We moved it through every doorway, and we got the results of what we have. We had a single purpose hypothesis, but now we're looking at a very robust platform. O19 is a strong neuroprotectant. It's also a potent anti-inflammatory. And you can see by the color coding in the schemes on this slide that those areas speak in neuroprotection, chronic pain, and mitochondrial dysfunction in refractory epilepsy, osteoarthritis, and Parkinson's. So we have a platform-based drugs, ladies and gentlemen. It's very drug-like. Bioavailability is high. I touched on it before. The inefficiencies of CBD are what they are. We penetrate the blood-brain barrier. We have 24-hour retention in blood plasma. We're going to be looking at that in terms of metabolites because we hit 2 biological targets of many that CBD hits, but we know that there's more specificity and less tracking in pleiotropic. So we're very, very comfortable with where we're at. Again, very drug-like, sub-nanomolar, globally patented, wide therapeutic index, really good stuff. I'm going to skip through these slides as quickly as I can. These are the 2 targets that we have focused on. KLS 13019 acts by helping restore mitochondria function and homeostasis. That's the NCX1 target for sodium calcium regulation. Very important target that plays a role in NLRP3 activation. Second is GPR55. It's an orphan putative cannabinoid receptor. It's coming very fast on the scene right now as a target that needs to be antagonized. And there's a reason for it. It's a bad actor, and its ligand, lysophosphatidylinositol, is also a bad actor. Very simply put, with chemotherapy-induced neuropathic pain, chemo increases the GPR55 target expression. We tone it down. We bring it back to control levels, and hypersensitivity and pain goes away. And the mechanism of action is known here in both GPR55 and in NCX1. Blocking pro-inflammatory targets is the key. Getting there early before inflammation starts on its time course is critical to preventing and reducing a neuropathic pain process. So we have a novel inflammatory target at molecular level and speaking to the inflammasome gene. This is some of the evidence that we have that we've been able to reverse The role of CIPN in the animal model. This is after the prevention. We've been proven to be preventative as well, but this is the home run money ball on reversal. You want to know that you can get reversal of neuropathic pain, and at the heart of it is inflammation. We're efficacious at 2 mg per kg in daily dosing and safe up to 300 mg per kg per Oz in vivo rotarod models. So rats just basically run around as if there's nothing going on. But the issue associated with the underlying problem is being dealt with. So toxicity is relatively low. This is the NLRP3 gene. You can see by just the graph alone that NLRP3 activation forced by paclitaxel-induced neuropathies focus on the targets of GPR55 and in NCX1. What's interesting here is the NLRP3 inflammasome detects danger signals and leads to release of cytokines. You know them all, IL-1B, big inflammation cytokine, and the killer from COVID is IL-6. And of course, TNF alpha is also well known. So we're able to get there before the real problem starts. And while even the problem starts, we're able to reverse it. These are the industry deals I talked about. You can only imagine who's next. I don't look at the market as a place where there's a lot of difficulty. I look at the market as a place where there's a lot of efficiency. You have to find the sweet spot in the market. And I think that we're in a good place at this time, leveraged with NIH money. This is what we're looking for. I've got a real opportunity. I'm happy to talk to anybody afterwards on a breakout to go over what this opportunity is and how it best leverages and opens up other market opportunities that I discussed in the program today. So with that, the appendix has other information. Everybody here is afforded an opportunity to get a copy of it, and I'll be happy to answer questions afterwards at the breakout session. Thank you very much.
Great presentation.
Thank you.
For being on time. If you wouldn't mind staying up here for a minute, we'll turn it to the panel.
Yeah, no
Yes,
problem.
I want to start us off with a few questions here, Dean. With the dual acting mechanism of action that you mentioned, and specifically the effect on both the central and the peripheral nervous system, what are potential off-target effects that you see for these therapeutics, including oxidative stress, even the potential affinity to opioid receptors or neurotoxicity?
That's a great question. Thanks for asking. On slide 19, there's going to be an evaluation to exactly answer that question. So we're free to give the slide presentation. But besides slide 19, NIH's preclinical screening program for pain ran some assay models, opiate assay models, safety net assay models, GABA-A receptor. They ran them as positive allosteric modulators in both the opiate and the GABA-A model. And in the safety net was the agonist antagonist model. And these were run at concentrations. The first two screens were run at 500 to 100, 5 to 100 nanomolar, 10 concentrations in duplicate. And the last of the GABA-A was run in the micromolar, rather nanomolar. I'm sorry, nanomolar was the first one, but the second was run, GABA-A was run in a micromolar range of 1 to 10 micromolar, again, in duplicate and across 10 concentrations. So we know that these results indicate that we have a clean compound with low inhibitory activity. It's not going to stop us from looking further down the line in ADME and other metabolites and drug and DDI interactions, stuff like that.
Thank you. Another question here is about the regulatory landscape. What does the regulatory landscape currently look like for these molecules being synthetic cannabinoids? And what is your strategy to address some of the challenges on the way to get to clinical trials and pathway to regulatory approval?
Another great question. Since we're considered cannabinoid therapeutics company, and we did look at CBD and we kind of angled ourselves off of that molecule, we wanted to test it against some of the real targets, CB1 receptor, CB2 receptor, and we do not have any significant interactions with either of those receptors, and we're structurally distinct from CBD, as you've seen on this slide with regards to the scaffold and the compound. It's not a controlled substance, so it offers the same challenges that other small molecule drugs offer when you go into the clinic. You have to go through the process of drug discovery and clinical evaluation. In order to address this question of abuse potential, we have 019 right now being studied at Temple University with Dr. Sarah Jane Ward, who's our principal investigator under the STTR grant. And she's looking at our compound in morphine and THC drug discrimination models. Thereafter, we have somebody lined up in year 2 and 3 of the grant. That's Ann Toland, who's at ATL Regulatory. And so she's going to be there looking to prepare us for a pre-IND meeting. So far today, we haven't seen an effect of O19 on herd channel interaction. CBD does have that interaction. So we're not so worried about QT interval and interruption for arrhythmias. CBD does have that problem. So when you go into the clinic and you're telling somebody you've got an analog that's better than CBD, you have to be prepared to tell them how different you are from that so that they don't stop you with preconceived notions of what CBD or it isn't. Beyond that, in concentrations and separation, we show the greater separation between active concentrations in nanomolar and CYP450 inhibition. So it should be less of a risk of drug interaction as compared to CBD. To conclude the thought, I would tell you that we're also looking at patient variability issues. We have a consultant online with us now, a fellow by the name of Nathaniel Katz, who runs Analgesic Solutions. And Nathaniel was an FDA guy, chief guy at the FDA, but he was running his own enterprise now at Analgesic to try to train and teach patients how to understand and expect pain, especially neuropathy in the face of chemo. So by using that patient interaction profile and selection prior to administration or patient registration, we could root out causes that cause variability in patients. And also we're working on a biomarker now so we can parallel up against the patient response. So we've done a lot of work to prepare ourselves for success in the clinic when we get there. We're not waiting to try and create a design. We're designing it as we go forward because we recognize some of the pitfalls.
Great. Thank you for that, Dean. So with that, I want to see if we have one more question from the audience.
Thank you.
What I'm wondering is, targeting the unknown is becoming very hard.
Thank you.
Why did you go for only neuropath? That's not it's a lot of issues.
Thank you for the question. There's an old Abbott and Costello skit where they're walking into a party like this and saying, we're going to eat it. And that's an example of something by accident. We first looked at a neurotoxicity in the brain years and years ago. So we knew that we could break blood-brain barrier penetration. So we knew we were active in the CNS, the hippocampal neurons. When we got into pain through CIPN, we realized we were in the peripheral nervous system. Now, both the CNS and PNS have active roles in NLRP3 regulation because the primary targets that we focus on, GPR55 and NCX1, are involved and they're everywhere. You can find GPR55 in blood leukocyte. I'm not suggesting that we're going to get burdened by Parkinson's or Alzheimer's. I want to be clear in the audience so that we know that we're only $1 million to run. I'm hearing from who's interested and has been talking with us is that there's a synoptic view of treating Parkinson's now. There's no silver bullet. So if we become adjunctive in therapeutic design because NCX1 deals with mitochondrial dysfunction, that's a path for us to be involved and still involved in NLRP3. In fact, they gave $1 million to Inflazome before Inflazome got bought out by Roche. They found us, we didn't find them. They looked at the literature that we had published in peer review, said we need to talk to you. So we're actively pursuing research and development with Northwestern, which is their research arm. But we're not limited to that, and we're not going to pinhole ourselves.
Yeah, that's the point I was trying to make, that you have opportunity.
Yes, indeed.
Great.
Great. Thank you, Dean.
Well, thank you very much.
Thank you.
So with that, I'm going to turn it over to Bill White to introduce our next presentation.
Thank you. I have the pleasure of introducing Sterile Therapeutics. Presenting today will be Manohar Kattikam, the CEO. I'd like you to think about three things with respect to this particular opportunity. First is the experience of the management team. Manohar actually you hear me okay? Okay. Manohar actually played a small role in the development of infliximab, Remicade, which we at Johnson & Johnson happen to know very well, and is one of only 10 products to achieve cumulatively over $100 billion in sales. So we got an experienced management team. The second thing that I'd like you to keep in mind is the team has a viable platform for launching these 3 assets into very, very strong and growing markets. And then the final thing is that they have a viable pathway into very, very large markets like irritable bowel disease, NASH, the 2 lead the 2 lead metabolic assets. If they can reach that aspirational place, would play in markets in excess of about $50 billion. And then they also have a opportunistic asset in oncology licensed in from one of the leading cancer centers in the US that would play in a $25 to $30 billion market. With that, I'll turn it over to Manaher.
You want to do the next one?
Okay. Thank you, Bill. Thanks for the kind introduction, and thank you all for attending this today and sharing with our product and companies and what we are doing today. So before I get to here to also would like to, before I introduce the company, I would like to introduce about myself in terms of this development and what I've been in the industry for about 25 years, mostly about 15 years in the big pharma, including the Centocor, which Bill introduced. So in addition to that, I've been an entrepreneur for 9 years. This is my 3rd company. I have 1 exit so far in the drug delivery system in a fast dissolving thin film market for prescription products. We have taken the product company from the concept to commercialization of 11 products in emerging markets, and I got an opportunity to exit in 2017. So this is my company, Stero Therapeutics. Right now, we're running it. This is the 5th year at this company. Most of the times, it is being me as a background in the pharmacist, where I'm a little away from the research and more in the development. So you see our products being more on the clinic side, less of the discovery implementation, more on the development side. So let me start with providing what's the strategy of the company. So as a grant introduced, there are 11,000 rare diseases, but its idea is we have to pursue a where we see that there is an opportunity to make it happen and available for patients. So that's actually a realistic consideration, which we have to pick a product where we have the potential that we can see and reach the patient. So with that intent in mind, we have selected products which have a path of an orphan disease, leveraging the orphan disease opportunity for speed to the market, less cost to develop it. So that's where we are again going back to my days in Centocor. And I joined in Centocor when we were seeing an indicator for $300 million market, Crohn's disease. That's the way it got approved. But everybody in the company knows this is for positioned for regulated for a rheumatoid arthritis. It's a $7 billion market. So that's a way, kind of a similar strategy. So we have all start with a rare disease, but carefully chosen a product and a disease where has a potential to expand to the bigger indication in the market. So all of our 3 products have that kind of a common characteristic, which is first one, ST002, is the fluoxetine. The inventor is sitting right here. Squad is the inventor of the molecule. So we positioned this. It has a clinical data of 120 patients so far, and we positioned it for Cushing's syndrome. We had a couple of rounds of discussion back and forth with the FDA, and we set this and agreed with the FDA for a phase 2 study based on the data that we already have. But it has a strong NASH data, which we have done in a dimensional model as well. Our second product is a peptide molecule, which is for primary sclerosing cholangitis. It's an orphan disease. It's a bilateral liver disease, and having a potential to expand into IPG. And the last molecule, which is being licensed from Moffitt Cancer Center in Florida, that is a multiple myeloma for and has the potential to have a data and availability for osteoarthritis. So where our products are, our product, as I said, first one, we are under NIH contract under part 3. We signed an agreement with Reda where NIH will conduct a phase 2 study for us, and we provide a GMP supplies. And we completed to the manufacturing. We are waiting for the IND to start and sorry. Okay. So, and that's where our phase 2. So we're going to start anytime. We are with the NIH collaboration under CREDA. The rest of the other 2 molecules we are going to file IND next year. So I want to give a little introduction of the disease. What is, many of you may already know it. It's a Cushing's syndrome, which is an excess cortisol secretion in the body. Right now, it is only a pituitary surgery is the first line of treatment. Only the drugs which are used for these patients is when there is a surgery which is not ineffective or not qualified for the surgery. And so in terms of what market opportunity, there are 25,000 patients diagnosed. Every year, there are 3,000 patients newly diagnosed for this. And there is a one benchmark drug, which is very controversial now, I guess, because it is a Korlym by Corcept. It is a repurposed morning after pill, mifepristone, positioned for this one. It gets a $175,000 reimbursement. So as far as the disease benchmark reimbursement is, we are in a very validated market. But because of some issues with the thing, market penetration never exceeded single digits. And here is our drug. We are targeting for this diagnosed Cushing's patients who are not qualified for a surgery or did not give a good result on the surgery. And we already have a FDA meeting with the regulatory presidents of a Korlym. We are using the same endpoint and same strategy for approval. We're moving on to our second disease, which is the liver bile duct disease, which is where you can see the scarring of the bile ducts of this. This is because of the excess cholangiocyte production. This disease is really debilitating, and there is no drug approved. It's an autoimmune disease with no alternatives except the liver transplant right now. So as far as the PSC market opportunity, there are 42,000 patients diagnosed, and there's no, as I said, it's no drug approved, and it has really the clear unmet need. It is the only alternative, as I said, is $600,000 liver transplant. With a 20% of the liver transplant reoccur the disease again. What we have a galanin 1 and 2 dual receptor antagonist. That's our peptide, where galanin produces excess or cholangiocyte proliferation, which is leading to the disease. And galanin does that proliferation. Our drug is a blocker of both the galanin 1 and galanin 2. In terms of the orphan designation, various status, both of our drugs, ST002 for Cushing's and PSC, both are approved, as orphan designations by US FDA in US. So we received orphan designations for both of them because it's a dual antagonist, and we got data to support a 24-week clinical trial when we move to the cell. We already have that data existing because this is the one which a collaboration with the Department of Veteran Affairs and University of Texas at Austin did extensive research, and before we licensed that from. Moving on to our last product into multiple myeloma. This is a bone disease most of you may be familiar with. This is dealing with a defective beta sites on that, and where we completely, those myeloma cells giving an excess secretion of metalloprotease 13, which is causing osteoclasts, which are the more bone cancer cells, you would say, at the cost of the good bone cells, which are osteoblasts. So excess proliferation of the osteoblast is what its disease is about and cancer is about. But our drug, which is being an inhibitor of this metalloprotease 13, is what we are approaching the disease right now. So as far as the disease prevalence, there are 150,000 myeloma patients. There are 12,000 deaths every year. Still, there are couple of therapies are already available, approved also, but still there's a 12,000 deaths every year. It is because either it is becoming resistant eventually by the treatments, available treatments, or they are toxic, or they're being very non-selective. So in that alternative right now, there is some help with the stem cell, but predominantly with the drugs and combination drugs, the outcome and the overall survival is very, very modest with available therapies right now. So why metaloprotease, in spite of many drugs are being there are many of it trailed because of the lack of selectivity for 13. That's the biggest reason. So we have IP, we have a global IP for all of these candidates, but we are mostly counting on market exclusivity with orphan designations. We already received 2 of them, as I mentioned. So the team, we are very well experienced in terms of our team is experienced in either in the small company setup like a biotech setup or big pharma setup, advancing the drug candidates through orphan designation. We are well experienced a team. We do have a scientific advisory team. It is very challenging to put a multi-therapeutic areas into putting scientific advisory board. We are still working on expanding it, but right now we do have the opinion given letters, which are very, very helpful for our program. So in summary, we have 3 drugs. 2 of them are already received orphan designation. All 3 are orphan diseases. We are positioned for a bigger indications once we get approvals for the smaller orphan diseases. We are currently raising about $5 million. Our current investors wanted to chip in part of that. And if anybody interested, I'm happy to address it, which makes, with this $5 million, we'll be able to take 1 molecule completion of the phase 2, others filing INDs for the rest of the 2. Thank you very much.
We have time for one question. Manhar, I wonder if for ST002, is there any additional information you could share around the discussions that you've had with the regulators that gives you optimism about a pathway to approval?
Yeah, so thank you. We completed 3 rounds at the FDA meeting with NIH, FDA, and the tripartite discussions, so 3 rounds. So because there is a regulatory precedence already for this indication, that was a clear, easy, and that's also our deciding point. So that consensus was very clear. And we had already 2 INDs with this molecule. So FDA already has the data on the safety point of view with this molecule. And studies were previously sponsored by NCI. So there is a good science and also good human experience of 120 patients. So they were very comfortable in letting us start phase 2 directly.
Okay, thank you.
We have time for one more question from the audience.
Sure.
So, this peptide that you plan to use, how are you going to deliver it to the liver?
So it is still a systemic way. It will be a first pass. We do not have any evidence so far that it will be chewed completely in the liver. So so far preclinical studies, we do not have any indication that it's disappearing, anything like that. We do have an efficacy in the preclinical studies going to the liver by straight intramuscular, by bypassing. No, so right now it's only intramuscular. IM.
Any other questions? We could probably fit in one more.
Sure.
This is all early because
Thank
an investor. It seems like you're splitting your focus
Yeah.
3 directions.
Yes.
And people like to say, oh, more shots on goal. I like to say lack of focus. And that means you're going to be moving the puck all around the same way, and you're never going to move forward. So why did you choose this approach rather than focusing on one, all your resources, all your finances, and moving one, charging ahead, and getting one?
Good question. Good that we thought about it. It's the first 2 products are metabolic diseases. So we have a common metabolic disease theme for the first 2 products. So only thing oncology product was an outlier too, except being a connection as an orphan disease. All three, we are an orphan disease company with all the three products being an orphan. Yes, it is a challenging. We have to pick the battle versus being an orphan disease company versus pick one orphan disease. But I wanted to be very be careful that we will be able to advance it. So our focus is right now on Cushing's ST002. We are having a discussions with the big pharma and some of the mid-sized pharmas about partnering the oncology asset because it does not fit into our metabolic profile. First two molecules are metabolic disease. But I agree the concern, but at the same time, we have to balance between those two.
All right.
Thank you very much.
And for our last presentation, I'm going to turn it over to Matthew to introduce.
Thank you. So the last company presenting today is Merlin Biotech, founded by Ian Henrich and Randy Heyer. This is a company bringing forward a mRNA platform technology with a pipeline, including a lead oncology indication. They have an experienced and innovative management team that will be moving these programs forward. Thanks, Randy.
Good afternoon. Hi. I'm Dr. Randy Heyer, and I'm the CEO of Merlin Biotech. Who here has heard of mRNA? Okay, just checking. And so we are a platform mRNA company. We are, now we talk about our assets, in particular our lead asset, an oncology asset. So the question I want to address today is why Merlin, why now? We do have this novel cancer therapy with human survival data confirmed in a mouse model. We have world-class leadership, experts, and board support. And we do have a potential for a large return on the investment in Merlin. Merlin was launched in April this year. Again, we are leveraging the mRNA technology for therapeutics and vaccines. We do have a robust pipeline with multiple shots on goal. I do want to focus in very much on our lead oncology asset, USP6, which has the in vivo data. Merlin is currently focusing nearly 99% of its effort on getting that product into the clinic. We believe that the problem, of course, is cancer, and which represents an enormous unmet need. We feel that Merlin's cancer therapy has enormous significance and value. The USP6 molecule is likely to potentially have an indication in a pediatric cancer, Ewing sarcoma, which is a rare disease, which is eligible for regulatory incentives, as well as accelerated development and potentially, upon approval, a priority review voucher, which is priced somewhere between $50 to $350 million, with the last two being sold this year, just a few months ago, for $100 million each. Adult cancer would also be a potential indication for USP6 with a broad application to numerous cancers, liver, pancreatic, breast, et cetera. And this could be co-developed along with the pediatric indication, more rapidly generating human safety and efficacy data to forward its approval. And there's potential for expansion in the adjuvant or the neoadjuvant space, as well as in combination with other cancer therapies. I want to introduce a little bit what is it? It's an mRNA-delivered cancer therapy. Here's a picture of a tumor cell. We use a syringe in the tumor to inject messenger RNA that encodes this protein called USP6, or ubiquitin-specific protease number 6, which is essentially a broad immunomodulator. Think of it as a rheostat in the immune system, and it turns up the immune system on three axes simultaneously, providing a very attractive opportunity to kill cancer. And that is, it helps recruit and stimulate immune cells. It activates killer immune cells and sensitizes the tumor to immune cancer killers. There's actually some human data looking at levels of USP6, it's a naturally occurring compound, in patient tumors that correlate with more favorable outcomes. And there's some data from Ewing sarcoma, and if you look at this graph, it's percent survivals on the y-axis, so higher is better, obviously. Those patients who had high USP6 expression compared to low USP expression clearly had a survival advantage in pediatric cancer, Ewing sarcoma, as well as in adult leukemia, the same effect. High USP was associated with survival. USP expression does segregate into bimodally, and it's either high or low with not a lot in between. So essentially what we want to do using our mRNA shred full of the USP6 is to turn people from low expressors to high expressors pharmacologically. And of course, this could go on to other cancers, including pancreatic, cervical, ovarian, bladder, breast, and so on. We do have in vivo data that confirms the human survival data. And this again is in a model, and we have child sarcoma or Ewing's on the left, adult leukemia on the right. Here's where tumors were experimentally introduced into mice, and we injected either no mRNA, a control mRNA in the green and black lines, or the USP6 encoding mRNA in the blue lines. And as you can see, for both the childhood sarcoma as well as the adult leukemia, we saw a distinct advantage with using active USP6 mRNA, thus confirming the human survival data. And then finally, which I find very interesting, there's an abscopal effect, which is a fancy term for when you inject one tumor with the USP6 mRNA, a distal tumor that was not injected also shrinks. So implying clearly a bloodborne means of transmission, clearly consistent with the mechanism of action as we understand it, and certainly a very positive step forward in our confidence behind the effect of the molecule. The data are shown on the right here. Those in blue represent the primary tumors that were injected with active USP6, and the effect on the distal tumor in blue, and the effect on the distal tumor in red for those primary tumors that were not injected with USP6. Cancer, of course, can be a very large market, and we believe that the USP market size can be driven by multiple applications to numerous cancers, indications, and expansions. This is incidence data in the United States for various solid tumors, obviously in very large numbers here, where USP6 being cancer agnostic would likely have an effect or an eventual indication in these tumors. In addition, as I mentioned earlier, there's a potential for an expansion to adjuvant or neoadjuvant settings, as well as combination with other cancer therapies such as CAR-T. The immuno-oncology rate, return on investment is quite attractive. This is a 2019 JAMA study. Median return was $14.50 per $1 invested. Median time to recover costs, 5 years. The market is large. The global cancer market is some $133 billion. With 33% of cancer drugs approved, netting over $1 billion in sales. So who is Merlin? Merlin is a very robust scientific advisory board. We have Harvey Alter, Nobel laureate in 2020, Stan Plotkin, a very preeminent vaccinologist, Margaret Chow, Professor Chopin, a co-inventor of the USP6 technology, Mike Mitchell, also downtown at Penn, an expert in LNP drug delivery technology, which might be very interesting as we try to expand indications in terms of steering the lipid nanoparticle. Chen Liu up at Yale, a liver cancer expert in pathology. Leadership, of course, starts with me, myself, as the Chief Executive Officer. I'm the former Senior Vice President of Global Medical at Moderna, very much involved in the approval and launch of the COVID-19 mRNA vaccines, as well as previously at Dynavax, for the FDA approval of Heplisav-B, a two-dose hepatitis B vaccine, as well as Jutao Guo, our Chief Scientific Officer, a co-inventor of some of the Merlin hepatitis B technology, a chief scientist here and a leading HPV scientist, as well as Ian Henrich. Ian is our VP of Oncology, an innovator, and a co-inventor of the Merlin USP technology. And we're supported generously by our board, led by Dr. Tim Block, their President Emeritus up here, Lauren Danzis. Thank you, Lauren. Outstanding business support, clearly a leader, as well as Carol Brogart, a clinical professor of medicine in San Francisco, many drug approvals under her belt, and I had worked closely with her in Heplisav-B. Financially, we're targeting an IND 2024 first half. We do have an academic entrepreneur grant in track for $650,000 with NIH applications that have been very favorably reviewed, scored, and with those represent $500,000. We are seeking $10 million in funding with a 20% equity stake. So why Merlin, why now? We do have the novel cancer therapy with human survival data confirmed in the mouse model. We have broad IP worldwide through licensing agreements with CHOP. We have a rich pipeline to follow it with multiple shots on goal. I do want to emphasize that all of our effort right now is very much focused on getting USP6 into the clinic. We have world-class leadership, experts and boards that are distinguished, innovative, and experienced with drug approvals under their belt, a track record, and the potential for a 10x return on investment with an IND potentially in 2024, and the potential indication for this molecule in numerous cancers, combo therapies, neoadjuvant, and adjuvant settings. Thank you.
All right, great presentation. We'll turn it to the panel first.
Yeah, two questions, Randy. Thank you for the presentation. I'll just echo some of Grant's comments at the start of today where, you know, pharma and potential M&A scenarios are looking for things that are diversified, developed, de-risked. Talk to me about the differentiated opportunity in USP6 expression versus current immunotherapies.
Sure. Well, I think USP6 offers quite a few. First of all, it has a broad stimulation of the immune system, both the innate and the adaptive response. It acts on a tumor simultaneously in 3 axes with sort of a 1-2-3 punch, as opposed to even a 1 or a 2 punch, by helping recruit immune cells, activating killer immune cells, increasing sensitivity to cancer killers. And it's very likely cancer agnostic. So it might work we anticipate that it might have an effect against all tumors, whereas many cancer therapies, particularly some of the more popular ones right now, require certain markers such as checkpoint inhibitors with a PD-L1 expression.
And then second question, intratumoral delivery. Can you comment on that approach?
Yes, I can. Certainly, also from personal experience at one of my previous companies, we were also developing an immuno-oncology in a phase 2 study with intratumoral delivery. It offers increased specificity, obviously, because we're delivering the agent directly to the tumor. And we know that it has an effect. And I did talk about that abscopal, or that remote effect. It localizes the expression, of course, of the cancer agent. And the expert opinion, as well as practical experience with intratumoral injection, is actually rather favorable. In addition, just if you think about it for a minute, if we administer a systemic cancer therapy, the toxicity profile, potential for toxicity is clearly much higher than a very localized, focused cancer therapy.
Thank you. Questions from the audience?
Yes, sir.
You talked about the abscopal effect. Is the primary and visible tumor in the bowel simultaneously, or do you take the primary tumor out and later on put the distal one?
No, they were not added subsequently. They were done at this. Tumors were installed. The one tumor was injected, the other tumor shrank with the primary tumor. And also that abscopal effect I've seen with the other intratumoral therapy I was working on, another immunomodulator. We see the same effect. You inject one tumor and you see the simultaneous or almost simultaneous shrinkage of a distal tumor.
Half your data you showed was with leukemia. Could you explain how you get intratumor injections in leukemia?
Yeah, that would be a development opportunity for us. I think what we're trying to show there is that the USP6 molecule is acting on different other tumors as well. Are primarily going to go after solid tumors in humans, as well as the pediatric in adults, as well as the pediatric solid tumor. A leukemia indication would be an expansion. We would have to modify. We'd have to do some more drug development to deliver the molecule. Right.
Do you know whether it has an effect on metastasis in sarcomas and solid tumors?
On the on the Mets, yes, we would anticipate that it would. I mean, the data that I showed you, the human data, survival data, that also includes a large number of patients that do have metastatic disease. So, if we believe the human survival data for the expression of USP6, that would confirm that. Yes.
So, expression of USP6 varies from patient to patient. How do you control
it? Perhaps, although I think that what we have to look at is when we look at the human epi data, the expression of USP6 falls into 2 large categories, low and high. So again, pharmacologically, we want to turn the low expressors into the high expressors. Now, there may, as we expand indications, there may be different levels of drug given for different types of tumors, maybe depending upon size and other things. We did a little bit of dose ranging before when I was doing the clinical work. So I can't answer your question specifically about personalized medicine. I just know that epidemiologically we see low and high right now. Our intention is to pharmacologically turn the lows into the highs.
Is there any danger of
None that I know of.
Dr. Hart,
Yes.
did you see anything that would give you an indication of how durable that response might be?
Well, we did see in some of the cases in the mouse experience, we did see complete remissions, the complete disappearance of the tumor. Now it's a mouse, so we can talk about that. So I don't know. The other thing, we are, if the molecule is safe and effective, there would be a potential for repeat administration, of course.
Yeah, great thought. You looked at USP6 gene mutations at all in terms of associated?
Yeah, no, we haven't.
All right, any other questions from the audience? Probably have time for one more. All right.
Yes.
This one is this do you know if there are specific methods or ways to deliver for
I just didn't hear your question.
let's say if I want to deliver
Sure.
look at the origin there and this way.
Of course. In fact, as I indicated in our advisory board, this is one of the areas of interest to us. The classic mRNA delivery mechanism at the moment is an intramuscular injection for a therapeutic vaccine. We do have this experimental data showing intratumoral injection, which of course is a very attractive way. I think as we would build, we would of course do additional research on finding other ways to deliver the molecule. One obvious thing is that this is effective in lung cancer. There would be a tremendous opportunity there and an interest in delivering it via to the lung. And I have some experience with that in the clinic from a previous application as well.
All right, riveting discussion. Thank you so much.
Yeah, my pleasure.
All right, so with that concludes our main segment, and I'm going to turn it back over to Lou for our Hall of Fame introductory ceremony.
Thanks, Grant. Thanks. I want to thank all three companies and three panelists and Grant. Great job, everybody. Really impressive. Before we invite the first speaker up, I just want to remind everyone that all three companies have a high top outside. So when we do the reception, if you want to ask more questions, anything else with investment, bring your checkbooks, but you can go right outside there. All three companies will be there. Secondly, I really want to thank all our sponsors. This would not happen without our sponsors. So we want to thank them. And with this time, this is a really special Wall of Fame induction. You can see when you go out, we have our FDA drug makers, and then we have our Wall of Fame with elected officials who've really been tremendous helps and impactful on our biotech center and on the community. So with that in mind, Senator Steve Sanasario is going to be inducted tonight. And we have two speakers who'd like to come up and say a few words. And then we have the senator. Hopefully our video and Zoom will work and we'll be ready to go with that. So our first speaker is the president. He is the chairman of the Pennsylvania Biotechnology Board. His name was up here earlier. Lauren Danzis to say a few words. Thank you.
Thanks. Well, where is Steve? Is Steve here yet? Okay, very good. All right. So 2 years ago, we broke ground on this $19 million expansion project. And of that amount, 4.4 million, or roughly 20%, was provided by the state of Pennsylvania. And that would not have been possible without the efforts of Steve and other supporters. We made a lot of progress and completed construction just about a year ago in October 2021. But we weren't done yet. We, during the course of that, had labs to complete and finishing work. We had space to build out. And so we called upon Steve for support, and Steve came through in major ways. First, there was a $1.5 million RACP funding grant in December 2021. What followed was a $722,000 grant in April of 2022. There was along the way COVID grants to support the development work being done by scientists at Bloomberg. And there were multiple grants to portfolio companies located here in the Biotech Center, including John Culp and Everest Bio, and a large grant of $750,000 to Flowmetric, which used that to purchase lab equipment and expand testing. These would not have been possible without the support of our advocates in Harrisburg, led by Steve Sanacero. We would still probably have a biotech center if Steve wasn't here to support us, but it would not have this audio system. It would not have this furniture, and it would probably not even have HVAC. I think we would be sitting in a sauna today were it not for Steve. Steve, thank you for what you've done for the Biotech Center. And beyond that as well, I know you're a huge supporter of the Cadence law, which is something that's near, as a foster parent, and near and dear to my heart. This is a young woman who was killed by her biological father during a state-sponsored visit. And Steve is working hard to change Pennsylvania law so that these visits are supervised. Thank you, Steve.
Next, I'd like to bring up our President Emeritus and the founder, and the reason why we're all here today to say a few words, Dr. Timothy Block.
Couldn't let an event go without me saying a few things. So, well, first of all, I'm sorry the senator can't be here personally today. I'll let him speak to that. But thank you for all coming, and thank you, Senator, for being there. So yes, as Lou mentioned, I retired from the leadership of the Biotech Center and our organizations just this month. But of course, I had to come back for this and come back for the spotlight, but also to thank and recognize Senator Sanacero for what he's done. Now, the Wall of Fame out there, which we moved, so it's a movable, but your photo won't go anywhere, Steve. It's going to go on the wall. We moved from the other lobby when we created this big building. Is out there and has a real place of honor and a great meaningfulness to us. As Lou mentioned, there's the Wall of Fame for those who've discovered FDA-approved drugs, and we have a few of those gentlemen in the room today. But then we also created a Wall of Fame for legislators elected who've done extraordinary transformational things for the Biotech Center. Now, that decision, who goes up on that wall, is an action of the board of directors of the Pennsylvania Biotech Center. And it's not something we do annually. It's something we do when there is a real reason to do it. And Senator Sanacero is certainly a reason to do it. And that's why I wanted to come back and be here to say these words about the senator. Now, Lauren mentioned some of the grants that the senator has gotten for us, but I also want to say some things about what Steve does actually in Harrisburg as really one of the most eloquent, forceful voices, not just for the PA Biotech Center, but for innovation, biotech, education, and of course the environment and consumer protection. And we've heard what he's done for individual rights. He is the chair of the Judiciary Committee. He is on the Appropriations Committee. He's on the Consumer Advocacy Committee and the Environment Committee, all in the state Senate in Harrisburg. He led our biotech think tank last year, and I assume we're going to do another one of those this year, where there was a brainstorming event which helped lead to some very influential actions in the state this year, an angel investment program, which of course we hope will benefit some of the companies here. Not everyone here may know what brought Steve to the state senator, to elected office. He, back 20 years ago, was a high-flying attorney with an office that had a view of the World Trade Center, and of course we know what he saw in 2001. So he was very moved by that horrific act in 2001, left that practice and got an education degree from Holy Family. So I meant to say that Steve obviously is eloquent and extremely highly educated, Tufts educated, with a law degree from the University of Pennsylvania. And so he was practicing law and watched what happened at the World Trade Center, was moved to become a school teacher and then taught in Bucks County. And after that was further moved to seek elected office in Harrisburg. And of course, we're very grateful that he did. So when we put someone on the wall out there, it's our way of recognizing them permanently for what they've done for us and for biotech. And that's why I feel so strongly about seeing Steve up there. And so congratulations to you, Steve, and thanks for what you do. And I see Rose is here from Steve's chief of staff, and thank you for what you do. So with that, I know we're going to do the unveiling and hear a few remarks from Steve. I don't know if you can see us. Oh, you can see me. Can you see the audience here, Steve? Oh, I'm going to come in.
The floor is yours, Senator.
Okay. Thank you, Lou. I hope you all can hear me. I want to thank Lauren and Tim for those very kind words, and I apologize that I can't be there today. Ronnie and I were actually looking very much forward to this and to being at the Shark Tank personally. Unfortunately, COVID had other plans for me, as seems to be the case for many people now in this month of July so far. Having said that, I am happy to report that I'm on the mend, and that is, I'm sure, in no small part due to the vaccines. Again, our thanks to people like Randy and many others who were involved in the development of the vaccines in record time, and the millions and millions of lives that they have saved across the globe over the last 2 years. And that, of course, brings us to the point of today, for me anyway, and that is just what an important role the Biotech Center plays in research and development. And for those of us locally, it can't be overstated. The success of the that Doc and Lou and Sherry and Lauren and so many others have had over the years it's really produced a our community, and fortunate to be the state senator here for the last 4 years, and to have this this organization in my district. It was a no-brainer for me to support it and to do everything I can to help it grow and continue its mission. And while I'm very grateful for this honor today, there are people who have stepped up to make it possible for the kind of support that the Biotech Center and the Blumberg Institute and the Hep B Foundation have received over the years. I, in particular, want to thank the Democratic chair of the Appropriations Committee in the Senate, Vince Hughes, his counterpart, chair there, and a gentleman who has become my friend over the last 4 years on the other side of the aisle, Pat Brown, who is the Republican chair of the Appropriations Committee in the Senate. Those three accepted my invitation a little over a year ago in March of 2021 to come out and actually visit the Biotech Center and to hear firsthand what the vision was there, and to kind of create a biotech corridor in our part of southeastern Pennsylvania. And to their credit, they got it almost immediately and became advocates as well for everything that you're doing and for these various grants that have been described. So I want to thank them. And of course, I want to thank Governor Wolf, who is, as you know, winding up 8 years as our governor here in Pennsylvania. And one of the hallmarks of his term in office, and I think one of his great legacies, is going to be his vision for Pennsylvania economically, of what we can do to really make our economy much more dynamic and attract people to the state. And in that regard, I just want to note, you may have read in the papers that one aspect of our recent budget deal completed in Harrisburg was something the governor proposed back in the winter time. In fact, something he's proposed just about every budget year in the last 8 years. And I was actually honored to introduce as legislation, and that is the cutting of our corporate net income tax, which right now at 9.99%. Under the deal that was passed in the next 10 years, that will go down to 4.99%. And that will really make Pennsylvania, over that time, it will be phased down over those 10 years, a much more competitive place, and will, I think, help us attract more business development here to PA. And that will help the Biotech Center and many other incubators across the state. So I'm proud of that. I'm proud of what we've been able to do so far. And I'm obviously honored by this this designation. But I want to say and make it clear that we're not done yet. There's a lot that is left to be done. Tim mentioned the biotech roundtable that we had last fall, and I do hope to continue that discussion. I think we're going to have a new governor come January, and a new legislature as well. We're going to be able to start getting real traction for a statewide dialogue about what we can do to make biotech a driver for our economy here in Pennsylvania, to support what we have already and to grow it even further. And that obviously, from my perspective, starts right here in Bucks County in the PA Biotech Center. So I want to thank, again, all of you for what you do on a regular basis. I have to say, I feel a little old this afternoon when I heard Grant Astor talk about Paul Volcker. I'm one of those folks who was around when he was the head of the Fed. So, but I have to say that it's, it is it's an honor for me. And again, I apologize that I can't be there with you today. I'm very grateful that we were able to get this this video link put together. So again, thank you very much.